Hello, welcome to the lab video. Tomorrow in class we're going to be doing a lab that has to do with how substances can diffuse through cells given their different sizes. Before we do that, we need to learn something about acids and bases that are going to be used in this lab. So I'm about to show you something which you will be able to see, and it will make sense of why things in this lab are going to change color. So what I have here is a indicator called phenylalanine, and I am going to pour some phenylalanine into this beaker that I have here. Now phenylalanine is an indicator. It turns colors in the presence of acid or base. So what I'm going to do is I'm going to take some of this base here, this is sodium hydroxide, and pour it in. You're going to see something's going to happen. And if you look, it's turned a bright shade of purple. So, we are going to be given, in the lab, you're going to be given these cubes, which I'm going to make the thing full screen, and these cubes are going to be this color, here, and they'll be this color because phenylalanine turns color in the presence of a base. Now, look what happens to phenylalanine when you pour an acid into the base. And I'm going to do it slowly so you can see what's going to happen. So here's I'm going to do it slowly. Now, if you look, what's going to happen? It's slowly but surely the substance goes back to being clear. So if you look here, it's now clear after pouring the acid into the base. So what we're going to do in the lab is you're going to be given different cubes. And these cubes are going to be this purple color because they have phenylalanine in a base. Okay? And then what's going to happen is you're going to stick these cubes, they're going to be different sizes. We're actually going to be using 1, 2, and 3. I don't think I'm going to be able, centimeters. I don't think the 4th centimeter cube is going to work out because the gels that I have are not big enough to make them. I'm going to pre-cut them for you to make the lab go faster. But we're going to stick these cubes in acid, okay? And what we're going to do is we're going to watch the acid diffuse through the different size cubes. If you look on the next slide that I have here, you'll see that the outside of the cube has turned white. Okay, and the reason why the outside of the cube has turned white is that the acid has diffused into the cube, making a phenylalanine turn white. Okay, so basically what we're going to do is we're going to have different sized cubes that are red, and we're going to put them into acid, and the acid is going to make them turn white, and we're going to measure, okay, how white they turn. I am now going to open the lab document to show you some things that are important in the... Okay, so the first thing I want you to do is, if you look at the text that's here in bold letters, is I want you to hypothesize which of the cubes, and we're not going to be doing the 4 centimeter cube, so you can cross it out. I'm only going to be doing 1 centimeter, 2 centimeter, 3 centimeter. Which cube will be able to best obtain nutrients from the environment, or best be able, in this case, the nutrient that we're adding is the acid. So which one will be able to best obtain the acid diffusion, which will have the best acid diffusion. And now we're going to take a look at the procedure and review the procedure and some things that are important to do in the lab. So I'm going to have the agar cubes pre-cut. Okay, so you'll get these cubes that are 1 centimeter long each side, 2 centimeters long on each side, and 3 centimeters long on each side. And then you're going to get a bottle of hydrochloric acid, and you're going to pour about 200 milliliters into a beaker, which I'm going to give you. Okay, then you are going to put all the 3 cubes, okay, that you have using a spoon. You're going to place them into the beaker, and they're going to stay in the beaker for 10 minutes. During that time, okay, you should watch them, and you should also periodically stir. Okay, now, while you're waiting the 10 minutes, you're not going to be doing nothing. Okay, during the 10 minutes, you're going to be doing calculations for each cube. Okay, if you look at the next page, for each cube, you're going to calculate the surface area, the volume, and the surface area to volume ratio. Okay, surface area, I'm going to type it in so you can see. Surface area of a cube, okay, is equal to 6s squared, where s is the side of the cube. Volume, I hope you know, of any cube is equal to length times width times height, but since the sides will be mostly equal, and yours may almost be equal, or the side of the cube cubed, or if it's not a perfect cube, length times width times height. So while your cubes are sitting in the acid solution, you are going to be calculating the surface area, the volume, and the surface area to volume ratio of your 3 cubes. You will not have cube 4. I don't have enough agar to make cube 4, so you'll only have 1, 2, 3, and 4. In fact, you should cross this column, this row out. You won't be using it. Okay, now, once 10 minutes has passed and you've done those calculations, you're going to use a spoon to remove the cubes, and you're going to place them on a paper towel and very, very gently pat them dry. Okay? You should very quickly get a photo capturing device, okay, and take a picture of the cubes. All this now has to be done quickly. Okay? Now, everyone should take 1 cube, okay, and you're going to measure the depth of diffusion. Okay, I'm going to show you what that means. The depth of diffusion is the distance from the beginning of the cube to all the distance that it's turned white to where it starts to turn red. So you're going to take a ruler, okay, and you're going to measure the distance, okay, from the end of the cube to where it is no longer white anymore. It has not turned white. That is how you're going to measure the distance diffused. Then, you are going to measure the length, width, and height of the area that's still pink. So if we go back to this picture, you're going to measure the length and height, actually not length, width, and height, just the, you're going to measure the length, width, and height, yes, the length, width, and height of this. It's going to appear 3D, you'll see it when you actually do it, the length, width, and height of the area that's pink. You'll see it will also go deep into the cube. This is just looking at it on top. So the length, width, and height, okay, of the volume, essentially, not the area, the volume of the area that's turned pink. Okay? If you're having trouble with this, you need to immediately call me over during the lab, because this needs to happen quickly, because even though the time is stopped, you're taking it out of the acid is still diffusing. So your goal is to measure 2 things. The distance from the beginning of the cube to the red, and so the distance from the beginning of the cube, okay, to the red, and then you're also going to want to measure the length, width, and height, okay, of the part that is still red. Those are the 2 things you're going to be measuring after you take the cubes out of the acid. Once you do that, all these measurements are going to go here. So we have the depth of diffusion, that's the distance from the white to the red. And then on the side, here you're going to calculate, in this area here, you're going to calculate the volume. So like the 1 centimeter cube, you'll put your length, width, and height here and multiply them. Okay. Then using those values, and we're going to talk about the rest of the calculations in class, because they're a little bit complicated. We're going to do some other calculations in class, including the rate of diffusion and the percent diffusion. Okay, but those are the calculations we're going to do more so in class than explaining now. So tomorrow, when you come to class, I want you to have your lab ready, and I want you to be with your group. The materials will almost already be on the table. Okay, so, and then as soon as the bell rings, I'll sort of give some last-minute instructions, and then you need to go. Hopefully, with all the time allowed in class, we'll be able to do the extra calculations, and you'll have time in class to answer these questions. If not, unfortunately, there'll be homework along with the regular homework in the lab. So the more quickly you do this, the less homework you're going to have. So I am now going to write a Google form so that I know that you watched this video and know what we're doing in the lab. And you need to answer the questions in the Google form. I'll send the address in the email with the link to this video. I will see you guys tomorrow. Be ready to do the lab, otherwise you're going to have an angry Miss Russo. Alright, have a good evening.